Students, in this video lecture, we will talk about recombinant DNA preparation, their transfer and their screening method. First, we will talk about recombinant DNA. So we will understand what is recombinant DNA. Bringing different genetic material from different sources together to create a sequence that would not otherwise be found in the nature. It means when we combine different DNA from different sources, then a whole combination will be called as recombinant DNA. Suppose we are taking DNA from bacteria and another DNA from human or plant, and we are joining both of the genetic material, then this construct will be called as recombinant DNA. Now we will see what are the steps which will be undertaken to prepare a recombinant DNA. The first step is isolation of genomic DNA from source organisms. Source organisms are the organisms of which we are going to get the protein or phenotype. So first we will isolate the genomic DNA. Then we need to get our gene of interest out of total genomic DNA. Then we will isolate the gene of interest from the genomic DNA. It means, suppose we want to express the growth hormone. Then we need to cut the DNA which is producing growth hormone. The stretch of DNA, or we can say the gene of growth hormone, we will isolate from the whole genomic DNA by utilizing restriction endonuclease, and the process is called as restriction digestion, and followed by isolation of gene from the agarose gel. Means in agarose gel we will isolate our gene of interest. We have already discussed this process in my previous lecture. And the third step is restriction digestion of vector. Means now we will digest the vector also. Vector is the vehicle which will transfer our gene of interest into the host. For example, plasmid. Now we will take the example of plasmid to explain this process. The fourth is ligation of gene of interest in a vector by ligase enzyme. So what we will do, we will just use a molecular glue, and the molecular glue is ligase enzyme. We will utilize ligase enzyme and we will fix our gene of interest into the plasmid. And fifth is introduction of recombinant DNA into their compatible host. Now by this schematic representation, we can understand all these steps by this figure. So we can see here that we have isolated this gene of interest, and for example, our gene of interest is growth hormone. It means if we want to express this growth hormone from the bacteria, then we will cut this gene of interest by using restriction enzyme, and we will isolate it in agarose gel. And after extraction, we will find this particular segment of DNA which transcribes and ultimately translates into growth hormone protein. Now we have a stretch of DNA out of total genomic DNA of the human. Now we will take here plasmid. As we know, the plasmid is the extra chromosomal genetic material of bacteria. Now we will take this plasmid as a vector, or we can say the vehicle. And now we will again use the restriction enzyme. After using this restriction enzyme, this plasmid will be opened. Now this plasmid, we can say it has been linearized. Now by using ligase enzyme, we can fix this segment of DNA from human into this plasmid. We can see here, this is the plasmid and this is the human DNA. So we can see here that we have prepared recombinant DNA, because this structure is having DNA from two different sources, one from bacteria and another from human. Now this recombinant DNA will be transferred into the compatible host. And in this case, the compatible host is bacterial cell. Now we can get the human growth hormone protein from this bacteria. There is no need to collect the growth hormone from human only. Now we will see different methods of transfer of recombinant DNA into the compatible host. So we utilize different methods to transfer this recombinant DNA in different hosts. So first method is direct method or temperature shock. This method has been employed to transfer the plasmid in bacterial cell. Now we can see here in this figure that we have taken the bacterial cell in this tube. In this tube, we have put this bacteria along with calcium chloride. Now we can see here that we have put this our recombinant DNA. We have prepared already. We will put this recombinant DNA along with calcium chloride with bacteria at 0 degree centigrade. Means we can put this, we can create this environment in ice. We will put all these things, recombinant DNA, calcium chloride and bacteria in ice. Then after some incubation, we will put this bacteria at 42 degree centigrade. So the bacterial cell will receive a shock. Firstly, the cold shock and suddenly the heat shock for 1 minute. Then what happens? The cold and heat shock will create several pores in the bacterial cell. Now we know that this bacteria is, this tube is containing bacteria along with recombinant DNA. So when the pores of the bacteria, including cell wall and cell membrane, opens, then our recombinant DNA will be inserted inside the bacterial cell. So by this method, we can see here that our recombinant DNA has been inserted inside the bacterial cell. And we should know that what is the role of calcium chloride. This calcium chloride and especially calcium ion helps the binding of DNA to the surface of bacterial cell, so that this calcium chloride or especially this divalent cation calcium will help, or we can say to increase the efficiency of this transfer of DNA into the bacterial cell, or we can say the transformation. Now this bacteria will receive this recombinant DNA. Then what we will do, we will grow this bacteria in this medium, and this nutrient medium, we will put all the colonies of this bacteria in this Petri plate. Now we will incubate this plate at 37 degrees centigrade, and after 8 to 10 hours we can see several colonies of bacteria will be grown. Now these bacteria will have our recombinant DNA. The second method is direct method or micro injection. Now we can see this is the cell and we are putting the recombinant DNA by an injection. This is a special kind of injection which is called as micro injection. Generally it comes with an instrument which is called as micro manipulator. So we can see here by this machine we hold this cell and we just insert this micro injection inside the nucleus, and we use to inject our recombinant DNA directly into the nucleus. But the person who is doing this experiment should be very expert. Now the injection of DNA or injection of DNA construct or RNA directly into the cell with the micro injection is being done, and generally this procedure has been used to insert the recombinant DNA in animal cells. The third is gene gun method. So we can see here that there is a gun-like structure which is having particle bullet, and this particle bullet is containing gold beads. So we used to coat our recombinant DNA with these gold or tungsten beads, and after a bombardment of this particle on directly onto the cell, what happens? This gold along with recombinant DNA directly will be inserted into the cell. Generally, this gene gun method has been employed to introduce the DNA in plant cells mostly. Now we will see the selection of recombinants. Means we will see in this, by this procedure, or we can say the method, we will see how and why we used to screen, we used to use this method for the screening purpose. So first what happens, there are two methods of selection of recombinant. One is antibiotic resistant and another is blue-white selection. So we will understand this experiment one by one, and after this method, we will understand why these methods are needed. So we can see here that we have taken the genomic DNA. After restriction digestion, we have isolated our gene of interest, and here this is the plasmid. After restriction digestion, we have cut the plasmid, and what will happen? We will use ligase. Then some of the plasmid will receive this gene of interest in this form. We can see here the plasmid with gene of interest, and some of the plasmid will also be there which will not be able to receive this gene of interest. Means it is called as empty plasmid, or we can say this self-circularized plasmid. So in this experiment, means after this, means during the formation of this recombinant DNA, two types of plasmids we can get. One is self-circularized or empty plasmid, and another is plasmid with our gene of interest. And this plasmid is of our interest. Means we need to get this construct in our host cell. Then what we will do, we will utilize one of the transformation methods. We have already discussed about the transformation method, so we will utilize one of them. And after transformation, we need to know that we can get three types of bacterial cells. We have, you can see here that there are two types of constructs, and both of these constructs will be transported into the bacterial cell. Then we will get three combinations of bacterial cells. Some of the bacteria will not receive or will not be transformed, and this bacteria will not contain any of the plasmid. It may be possible. Some of the bacteria will have the empty plasmid, and some of the bacteria will have plasmid with our gene of interest. So A has been denoted the bacterial cell with plasmid plus gene of interest. B is representing bacteria with empty plasmid, and C is representing empty bacteria. Means this bacteria has not received any plasmid. Now the mixture of this bacteria will be grown in a nutrient medium which will contain ampicillin and tetracycline. So we have utilized two different antibiotics. So now we will see the structure of this plasmid. Now we can see there are two antibiotic resistant genes. We have already talked about this antibiotic resistant gene. So we can see here one is tetracycline resistant gene and this one is ampicillin resistant gene. So these resistant gene helps the bacteria to protect by the application of these antibiotics. It means the bacteria which is having this plasmid will be protected by the treatment of either tetracycline or ampicillin or both. So we can see here that this bacteria will die, means the population of bacteria which is not having this plasmid will not be uh, resistant to that particular antibiotic. So all these bacteria will be killed. It means in this nutrient medium, this kind of bacteria will be deleted or will not be grown. Now we have to screen A from B type of colony. We need A type of bacteria. So what will happen? We can see here that there are two different genes for two different antibiotic resistant genes. So one is tetracycline and another one is ampicillin. Now we can see here that our gene of interest has been inserted inside this tetracycline gene. Now we know that the construct of this gene has been disturbed just because of insertion of our gene of interest. So this condition has been called as insertional inactivation. It means now this, after insertion of our gene of interest inside this tetracycline resistant gene, the construct of or sequence of gene of tetracycline will be disturbed, and this gene will no longer be functional. It means it will not be able to produce tetracycline. Now what will happen? We will grow this colony in ampicillin medium. Then what will happen? We will make the replica copy of this plate. Now we will grow this bacteria into tetracycline. Now we can see that in tetracycline, only B type of cells will grow just because they are producing both of the proteins, tetracycline and ampicillin. But A type of colony will not be grown just because of presence of tetracycline gene, because now due to insertion of this gene, this bacteria will not be able to grow in tetracycline medium. That's why we can see here we have already made the replica copy of this colonies of this bacteria. So we will compare now the dyed bacteria. When we will compare this plate with this plate, then the dyed bacteria after application of this tetracycline will be our interest of bacteria. It means we will select A type bacteria from our previous plate which has been treated with ampicillin only, and we will isolate, so we will get pure colony or pure bacteria of the construct with plasmid with our gene of interest. So by this procedure we can screen out those bacteria which is having plasmid along with our gene of interest. The second method is blue-white selection. Now we can see here the structure of this plasmid is somewhat different as compared to previous one. Here in the previous selection method, there was a gene which was a tetracycline resistant gene. Now this gene is LAC-Z gene, and this LAC-Z gene is producing beta galactosidase, and there is an ampicillin resistant gene also here. So this LAC-Z gene is producing beta galactosidase, and this beta galactosidase is a protein which reacts with a substrate X-gal. X-gal is a substrate which reacts with the protein of this LAC-Z gene, and it gives blue color. Now the same theory will be repeated here as insertional inactivation. Now we can see here that we have used restriction enzyme to digest the DNA. We have got this segment of DNA, which will be called as our gene of interest. Suppose it is a growth hormone sequence, and we have linearized this plasmid. Now we will ligate both of these segment of DNA, means plasmid and our gene of interest. Now again we will get two types of plasmid. One is self-circularized or empty plasmid, and another is plasmid with our gene of interest. Now again transformation will be done. Now we will employ X-gal on the surface of this nutrient medium. So we can see here that again this LAGZ gene has been disturbed just because of insertion of our gene of interest. We have already inserted the gene of interest in this particular plasmid. That's why now this plasmid will not be able to produce beta galactosidase. And if this LAGZ gene is not producing beta galactosidase, then what will happen? It will not be able to react with X-gal, and it will not give the blue color. So the bacteria with this plasmid will grow in white color. On the other hand, we can see some of the bacteria like this, B, A, B, C. So C, we can see here that they will be killed by application of ampicillin. Means if we will use ampicillin, then this bacteria, empty bacteria, which has not been transformed, will die. All the bacteria without this plasmid will die. So we have screened out this bacteria. This option has been deleted now. Now there is the condition of growth of two types of bacteria. This one means plasmid with our gene of interest and empty plasmid. Now we will talk or we will see the condition in this plasmid. So this plasmid, in this plasmid, the LAGZ gene is intact because none of the gene has been inserted inside this LAGZ construct. So it will continuously produce beta galactosidase. Now this beta galactosidase will react with X-gal, which is the substrate, and we have already employed this X-gal on this plate. So the colonies of bacteria which is having empty plasmid will grow in blue color. So now we will select the white colony only. So we can see what is going on here as this figure. We can see this was our vector, and some of the vector is self-ligated and some of the vector is will receive our gene of interest. So we can see here, this is the plasmid with gene of interest and this is the empty plasmid. Now, since this LAGZ gene is intact, so it will produce beta galactosidase and it will react with X-gal and it will grow in blue colony. Now we can see here blue, and now we can see here that the plasmid with our interest will not be able to produce beta galactosidase. That's why beta galactoside will not be produced, and this, if the beta galactosidase is not present here, so there is no chance of reaction of beta galactosidase with X-gal. So the colonies will appear in white color. Now we will select the white colony. So the white colony will contain the bacteria with plasmid with our gene of interest. Now we can utilize this bacteria to produce our product of our gene of interest, such as growth hormone or insulin or any enzyme or any protein. Thank you.